Welcome to the Apron Academy. This video is specially designed for the dietitian in training, also known as the RD to be. Today we're going to be talking about vitamin C. So this is a pretty popular vitamin, but we are going to talk about some stuff that goes on behind the scenes, but is so important in the function of vitamin C. So first, of course, we have all of the fruits, and I think this little GIF here was supposed to have oranges, which when you think of vitamin C, like I think of oranges, I think of red bell peppers, lots of citrus fruits, but fruits and vegetables is where we're going to find our vitamin C. So, it is interesting with all the citrus fruits that do have a lot of vitamin C. This is because vitamin C is really stable in low pH environments. So those acidic environments, vitamin C is all about that. So I want to reveal this little mechanism. So I've written this little mechanism. We have glucose, and then we get to L-ascorbate, which is our vitamin C. And there's a mechanism. I have done this before where there's a lot more going on than I'm writing about. But what we need to know is in order to get vitamin C, we start with glucose, which is really interesting. We have our glucose, and then we need this final enzyme, gallonolactate oxidase, in order to make this vitamin C, the L-ascorbate, in our bodies. So almost all living things can make their own vitamin C, but we as humans cannot. Because we don't have this gallonolactate oxidase enzyme. So this makes it really hard to study vitamin C in the lab because mice and rats, which are usually what's studied, they can make their own vitamin C. So they're not a similar type to us. But luckily, guinea pigs and primates and some birds, they are not able to make their own vitamin C, so we're able to have tests in that way. So, since we don't have this enzyme, the gallonolactate oxidase, we have to make sure we're eating all of the fruits and the vegetables. Dun, dun, dun. I turn the page or I scroll down and it's scary. We're going to break this down, but this is simply the mechanism of action for vitamin C. So by knowing this mechanism, we can know how vitamin C demonstrates its function. So we start out with ascorbic acid. This is similar to this L-ascorbate. We have ascorbic acid, and it's getting oxidized, and it becomes a ascorbyl radical. What happens? We're losing hydrogens and electrons. This OH group now becomes a radical. So this is really interesting because this ascorbyl radical can be oxidized and it can reduce other stuff without becoming toxic. So it doesn't damage other molecules. It's really stable. Oftentimes when we think of radicals, we think of very unstable, very dangerous or toxic. So the fact that this ascorbyl radical is stable and is able to reduce other things without becoming toxic is a very big deal. It's also a strong antioxidant, so it functions as an antioxidant and also as a free radical scavenger. So then this ascorbyl radical oxidizes and we get dihydroascorbic acid. So each time it oxidizes twice in this mechanism. Each time it becomes oxidized, it's reducing something else. So then I want to note that when we get to this dihydroascorbic acid, it's great to have vitamin C, but we are going to be stuck in this position if we don't have glutathione. So this is the next antioxidant. Glutathione is the most abundant antioxidant that's found in the cells. So using dihydroascorbate reductase, this is able to go back to the ascorbic acid. So vitamin C is, again, able to be used. I want to note that I didn't leave a whole lot of room over here, but sometimes a big, almost, I guess, buzz, news topic in nutrition can be that high vitamin C levels leads to kidney stones. So how this is seen is this goes to oxalate, and I forgot an A, but then it goes, it creates the kidney stones. So this does happen when we have extremely high vitamin C or someone is predisposed to kidney problems. But vitamin C can be excreted really well through the kidneys, so oftentimes this doesn't just happen just because. And it's the oxalate that creates those kidney stones. But I'm going to erase that for now. This mechanism is important to know because this is how vitamin C is going to be functioning in the rest of the body. So if we know this, that it's able to get oxidized and reduce other things, then we know basically the foundation of what vitamin C does. So how do we get it into the body? We have two transporters, SVCT1 and SVCT2. Now, sometimes in my videos I have an image of a little anatomical dude. We see his esophagus all the way through the gut, and then I explained that if we look into his intestines, this is what we'll see. So imagine we're in the intestines. This is actually the lining of the intestines, and so these are the villi. So this is going into an enterocyte, or one of those intestine cells in the lining. So where these transporters are located, this SVCT1 is located on this luminal side, and the SVCT2 is located more on like the blood side. This is how it will get into the blood. So both of these transporters work specifically for ascorbate. SVCT1 is expressed in the liver, kidney, lung, ovary, prostate, placenta, etc. It's also responsible for intestinal and renal absorption. SVCT2 is expressed by all cell types except for the red blood cells, but it's not expressed at the same level. So an important thing with the SVCT2 is when there is increased oxidative damage, there are more SVCT2 transporters that come to the rescue to reduce whatever is there for that oxidative damage. And this transporter is a redox sensitive transporter. So both of these are absorbed in the intestines. Both can prevent scurvy that I will talk about before, and these both primarily work for ascorbate. So dehydroascorbic acid, this one here, it also needs to be transported. Say we don't get all of that recycled, we still need to function with this. So dehydroascorbic acid is not transferred or transported by either of these transporters. It's transported via a GLUT transporter. So then once it gets into the cell, the dehydroascorbic acid is then reduced to ascorbate. So I want to talk a little bit about the bioavailability. The bioavailability decreases as higher doses of vitamin C are ingested due to the inhibition of those vitamin C transporters. So what does that actually mean? So imagine this is the amounts in units of how much vitamin C that we're getting. If we continue to have higher and higher doses, eventually our transporters, they're going to downregulate or shut down so that all of the vitamin C that we're ingesting when we get into these higher levels is really not getting absorbed. So we can see there's a huge difference if we, have at least a little bit of vitamin C. Like we see all this change. But when we get over here into the major amounts of vitamin C, there's really not a change. It starts to plateau. So, that's what I need to say about that. High concentrations of vitamin C are able to be dispersed throughout all the tissues. So, when we perhaps are deficient, we will lose those concentrations at different rates. So that goes back to this SVCT2, how it's expressed in all the cell types except the red blood cells, but not expressed at the same level. So we'll lose those concentrations at different rates if we're deficient. And typically the brain and the eyes are last to be depleted of the vitamin C. And so now let's talk about the function. We talked about the mechanism before, and that displays the mechanism of action. So that's what's going on. But I just want to explain some areas where it is specifically functioning. Overall, I want to mention that vitamin C is functioning as a reducing agent. So if we go back to this main mechanism, it's a reducing agent. It's getting oxidized itself, but when it works, it's going to reduce other things. Also known as an electron donor, where it hands off these hydrogens and electrons. So there are two large categories of functions. We have enzymatic functions of vitamin C and non-enzymatic functions. So first I'm going to talk about the enzymatic functions. We have mono-oxygenases and dioxygenases, and then I'll talk about the non-enzymatic functions afterwards. So first, mono-oxygenases, we're even going to break it down even further. So we have dopamine beta hydrolase. So it's this enzyme here. In this reaction, copper is getting reduced by the vitamin C, and you can almost imagine that big mechanism that was above is going on right here to help reduce this copper. So it functions here to be able to swap this dopamine to norepinephrine. And another monooxygenase is this big long name, also known as PAM, and it also is reducing the copper. Now we're going to talk about dioxygenases. So one of the biggest functions that vitamin C plays is its role in collagen synthesis. So I'm going to stop here for just a second. Vitamin C is so important for collagen synthesis. In fact, most of the important dioxygenases are work as the hydrolases in this collagen synthesis function. So when we have vitamin C deficiency, we're going to see poor wound healing. This is directly related to this collagen synthesis. So we see for the collagen synthesis, what's necessary is we need two enzymes that function in this collagen synthesis. The first is the lysyl hydrolases and the prolyl hydrolases. So proline and lysine. They function to hydroxylate the amino acids to form bonds between the strands and to strengthen the collagen. So what does that mean? We need to make these bonds. So this is a P. This all is representing the lysines and the prolines. So we hydroxylate it. We create these bonds, and that strengthens these strands of collagen. So now say this is making a bond. And when we continue to do this, and say we even do it up here, make that bond and that bond, it makes that collagen even tighter. Whereas if it was just, floating by itself or whatever, now it's very tight and is able to heal the wounds. So for this, in this process, vitamin C is needed to reduce iron. So that is how vitamin C plays a role in this very important function. Next, we have the HIF-1 alpha, also known as the hypoxia-inducible factor 1 alpha. It functions as an oxygen sensor. So it gets hydroxylated, And when proline is hydroxylated, this HIF-1 alpha gets destroyed. So in normal oxygen levels, it functions normally. Hypoxia, which is what this H stands for, means there's no oxygen. So this HIF-1 alpha, in that hypoxia, no oxygen state, is not broken down. It's stabilized, and it acts as a transcription factor that moves to the DNA and drives expression in the genes, specifically in the genes that we require when we don't have oxygen. So it responds with angiogenesis, so more blood vessels. In order to bring in more oxygen, we have glycolysis and pretty much survival. Thirdly, as a deoxygenase, we have fatty acids. So in addition to a vitamin C deficiency leading to poor wound healing, like we discussed in this collagen synthesis, vitamin C deficiency can also result in fatigue that's caused by the reduced synthesis of carnitine. So that is a key word for this one, carnitine. Carnitine is a carrier molecule for long-chain fatty acids in mitochondria. The enzymes require iron as a cofactor. We need vitamin C to oxidize. If no vitamin C is in this beta oxidation, then we won't be able to generate the energy from fats. So that's why we have, with a deficiency, lethargy, tiredness, and fatigue. So the synthesis of carnitine requires vitamin C to reduce iron as a cofactor. So I know I'm sounding redundant a little bit, but all of this is so important in the function of vitamin C. And last but not least, we have tyrosine catabolism. So one of the enzymes here, and I'm probably going to butcher the name, tetrahydrobiopterin. That actually was pretty good. This cofactor needs to be reduced by vitamin C in this reaction. So vitamin C is needed not only in these enzymatic reactions, but also in non-enzymatic functions. So I'm just going to talk about 4 here. First, it's needed for iron absorption. So vitamin C is used to increase non-heme iron absorption. Our bodies must absorb iron in its reduced state. We can't just absorb it like free iron when it's oxidized. So we need vitamin C to reduce it. Just want to point out that when someone is anemic, it is going to be very good, beneficial to use vitamin C in those anemic patients. But there are some cases when people can have too much iron, so it's important to pay close attention to the meals, especially if the meals contain lots of iron and vitamin C in the same meal. We can see hemochromatosis. All right, and so our next non-enzymatic functions, nitrates and nitrites, often these can be harmful substances to our body, but vitamin C reduces these so that it's a less harmful product. Vitamin C also acts as an antioxidant. So I talked about how it functions as a free radical scavenger and an antioxidant at the beginning of this video. It helps to reduce the reactive oxygen species. So vitamin E is another antioxidant vitamin. and it works alongside of the vitamin E. So we see in this reaction, we have vitamin E, we have this glutathione here, and this might look familiar. This is our NADP, our niacin. So nothing works in isolation. We're going to see this more as we continue to build on our vitamins, but this is just important to know, and know that it works as an antioxidant. Not only is it an antioxidant, it's also working as a pro-oxidant. So at very, very, very high levels of vitamin C, this takes place. Vitamin C in the presence of a cancer cell can become a pro-oxidant. So hydrogen peroxide is produced, helps to kill those cancer cells. So it's almost like a protective barrier when we do have those high, very high levels of vitamin C. Chemotherapies have also been looked into with vitamin C for its oxidative functions. Now, what happens if I don't have enough? I've written it here. Our deficiency disorder is scurvy. So scurvy can develop within 30 days since we're able to get rid of vitamin C quickly and not store a whole lot. Signs of this include coiled hair, bleeding gums, joint pain, etc. And this is directly related to our lack of collagen synthesis that we talked about above. Also due to the loss of those prolyl and lysyl hydrolases. So here we also have impaired carnitine and neurotransmitters, which results in fatigue and even depression. So there is a connection between the amount of vitamin C in the blood and decreased heart disease, stroke, cancer, cataracts, gout. So vitamin C is so important. It's even used to treat high blood pressure because it helps with the vasodilation we saw above, helps with wound healing, that fatigue. And it's really not toxic, except you can get to levels where you have osmotic diarrhea, so just be mindful of that. But really, we need our vitamin C. We need to make sure that we're taking advantage of all the good things that vitamin C does for us. If you enjoyed this video, please like and subscribe, and thanks for joining me.